Hello, good morning, everyone. You are welcome to BCHM 262 tutorial class. Before the tutorial questions, we are going to have a look into ketone bodies synthesis, as we've already discussed in the class, and also have an overlook on biosynthesis of cholesterol and the fat. So on ketone body synthesis, I have already told you the conditions that warrant the synthesis of ketone bodies, ketogenesis, where I told you in a situation where we have high fatty acid oxidation, and we have high acetyl-CoA, and when there is low glucose, some of these fatty acids, like I said, are used for energy production, quite a number of organs, but some certain organs prefer to use ketone bodies instead because of glucose is not available. They prefer actually glucose, and due to the fact that glucose is not available, and they prefer the use of ketone bodies instead of fatty acids, like brain we've mentioned, and some other organs. So those ketone bodies are produced in the liver. Specifically, they are produced in the mitochondria, and once produced, they then transported to brain cells, utilized for energy production. So how are these ketone bodies produced? Like I mentioned, the ketone bodies, usually we mention three: acetone, acetoacetate, and dihydroxy beta hydroxybutyrate. And we have mentioned that the reactions involve condensation of 2-acetyl-CoA by the enzyme thiolase. And remember, this enzyme we talk about in fatty acid oxidation, which is the last enzyme in the fatty acid oxidation, the cycle, thiolysis. And the next one is another acetyl-CoA coming to condense with the form acetoacetyl-CoA to give HMG-CoA. And this HMG-CoA formation, we'll talk about it while discussing cholesterol, or when you read the cholesterol metabolism, even though I will quickly go over the synthesis of cholesterol and see how HMG-CoA is produced, which is similar to this step of reactions. Two-step reactions are similar in ketogenesis and cholesterol synthesis. Now the next after formation of HMG-CoA is lysis or lysis of HMG-CoA by HMG-CoA layers, which acetyl-CoA is removed. Remember, the previous step involved the addition of acetyl-CoA, and the next now is the removal of acetyl-CoA. The only difference is that the addition of acetyl, initially it was to acetyl-CoA with a CoA, then addition of acetyl-CoA removed that first, it removed the first CoA, and then the acetyl-CoA added. Now the HMG-CoA only have one acid coenzyme E group. Then the next reaction is to remove acetyl-CoA. So all the CoA were now removed. What remain is acetoacetate, and acetoacetate, like we said, is the first ketone body produced. Now the acetoacetate can undergo decarboxylation reaction using acetoacetate decarboxylase, or it can spontaneously, without the aid of this enzyme, also can undergo decarboxylation to form acetone. And equally, another enzyme, beta hydroxybutyrate, can actually break down acetoacetate to give beta hydroxybutyrate. I remember what we said about beta hydroxybutyrate in the case of ruminant animal, where we said in ruminant animal, normally in most cases, because this one you have to remember, that in digestion of lipids and even in the digestion of carbohydrate, where I mentioned that these intestinal microbes, the microorganisms, that actually act on even the glucose itself, and these fatty acids, the long chain fatty acid, even broken down into smaller chain fatty acids. Remember the volatile fatty acids, and butyrate or butyric acid is one of these volatile fatty acids, the smaller chain fatty acids. So intestinal microbes in the rumen actually break down some of these even long chain fatty acids to these smaller chains. So in the rumen we have high concentration of this butyrate as part of the volatile fatty acids produced. So, and this butyrate easily absorbed, and when they are absorbed, they can easily be converted to beta hydroxybutyrate. So normally in a normal or single stomach animal, so even in humans, the concentration of beta hydroxybutyrate as the ketone body is at a certain concentration, whereas in the case of rumen we have a higher concentration of this beta hydroxybutyrate because of the simple hydroxylation reactions that lead to accumulation of this beta hydroxybutyrate, and we know the way they need to repeat also. Now the next thing is how we didn't discuss in the previous class, how this ketone body is actually produced or utilized in the body. And the utilization is starting with beta hydroxybutyrate, and reversal of the reaction of beta hydroxybutyrate dehydrogenase lead to formation of acetoacetate, and acetoacetate will now acquire the coenzyme E from succinyl-CoA. Succinyl-CoA has a coenzyme A attached to it. So removal of coenzyme A from succinyl-CoA by ketoacyl transferase will now transfer the coenzyme A to acetoacetate, forming acetoacetyl-CoA, and that same enzyme thiolase will now break down the acetoacetyl-CoA with the addition of coenzyme A to form 2-acetyl-CoA. So these 2-acetyl-CoA will be used now for energy production through TCA cycle. So this is production and utilization of ketone bodies. Now the next thing we talk about the cholesterol synthesis, but this one will be briefly, and the most important thing here to know is that cholesterol is 27 carbon molecules, and they are all synthesized from acetyl-CoA. But how are they synthesized? We are going to see. Now these are just some few organs where these things can be synthesized, and the enzyme is actually the combination of cytosol and the endoplasmic reticulum. We talk about 1-2 step where you see the next step involved an enzyme from endoplasmic reticulum, and the enzyme specifically here is the synthesis of mevalonate. The synthesis of mevalonate actually takes place in the endoplasmic reticulum. So we will see the steps, and these are the requirements. Remember, this is a biosynthetic pathway, where we said in biosynthetic pathway the reducing equivalent use is NADPH. So we have 16 NADPH we use. This 16 NADPH comes from, you know where the NADPH can actually come from. Most importantly here, almost particularly comes from the hexose monophosphate shunt, and 36 ATP is used. It's highly energy requiring process, and then the acetyl-CoA as the metabolic precursor, 18 acetyl-CoA, which comes from of course glucose metabolism. And the role of cholesterol, we don't need to keep mentioning cholesterol in cell membrane synthesis, steroid hormones, synthesis of vitamin D and lipoproteins, as well as in the bile acid. Remember we said something about bile acids, and this what do you call it, this synthesis or metabolism of cholesterol, which involve the formation of cholesterol and the breakdown of cholesterol. The lipids digestion and absorption actually depend on this metabolism, because bile acid play a very important role in the digestion and absorption. So how is it bile acid actually comes from? It's through the degradation of cholesterol. So the cholesterol degraded produce bile acids, and this bile acid play a vital role in what? In the digestion and absorption of these lipids. So without production of bile acid, and remember, even when we talk about digestion, we are talking about digestion. It's not all these digested lipids that actually, or it's not all the lipids taken digested and or absorbed. What facilitates digestion and absorption is this presence of bile acids. So we can see how bile acid play a very important role when it comes to lipid metabolisms. And when we say the whole lipid metabolism, or the digestion and absorption of lipid also depend on metabolism of lipid. That's something we have to corroborate the two information. So these are the sources of cholesterol high foods. Although we say that 80% of the cholesterol actually produced in the liver, they are endogenously produced in the liver, whereas only 20% is actually taken in. Only in the case of in digestive tract, only less than 50 or 50% of it actually taken in by during absorption. So these are the foods that have a high source of cholesterol. But remember, only 50% is taken actually been absorbed. So the remaining 50% or even 80%, only 80% actually this cholesterol are produced by the body. So this is just an overview of the synthesis. Like I said, the acetyl-CoA, which we have 3 acetyl-CoA forming HMG-CoA, and this is what we say is similar to in the case of ketogenesis. Ketogenesis, we have HMG-CoA, and the next reaction is HMG-CoA lyase. But here, what we have is HMG-CoA reductase that leads to formation of mevalonate. From mevalonate, then we have the isoprene unit. This activated 5-carbon and activated 10-carbon, activated 15-carbon, and the 2 activated 15-carbon will come together to give squalene, which is a linear 30-carbon, and then the rearrangement and cyclization form lanosterol. And after lanosterol, we have about 19 step reaction in the formation of cholesterol. And lanosterol is 30-carbon is still this 19 step reaction leading to formation of cholesterol involve the carboxylation reaction. 2 carbons were removed along the process, and of course some reduction reaction. So this is just some pet of cholesterol, steroids, bile acids, vitamin, etc. And we can see how some of the intermediates in the synthesis actually used for the synthesis of so many things, including carotenoids and the ubiquinones. Remember ubiquinones and plastoquinone. Ubiquinone specifically, we discussed about it, ubiquinone, in this class. And plastoquinone is something maybe you may come across subsequently, but it is particularly while talking about this electron transfer in plant, does we talk about ubiquinone. So vitamin K, E, is water soluble vitamin, lipid soluble vitamin A, D, E, K, etc. Now cholesterol synthesis, this, like I said, is the formation of HMG-CoA, which is similar to what we've seen in ketogenesis. And from there we now talk about HMG-CoA reductase reaction, where we have the reduction of HMG-CoA to mevalonate, and mevalonate, is a 6-carbon molecule. Now the next reaction is more of the step of phosphorylation reaction and the carboxylation reaction, like a 4-step. Mevalonate will be phosphorylated to form 5-phosphomevalonate, and then another phosphorylation at the same carbon 5 to form 5-pyrophosphomevalonate, and another kinase in the at position 3 of mevalonate to find 3-phospho-5-pyrophosphomevalonate, and then the next reaction, which involves the carboxylation reaction to form what we call isofentanyl pyrophosphate. So this isofentanyl pyrophosphate, this is the first 5-carbon molecule, and this isofentanyl pyrophosphate exists in equilibrium with its isomers, because immediately it is formed, it is isomerized, so it forms an equilibrium with the isomer, which is dimethyl allyl pyrophosphate. So dimethyl allyl pyrophosphate and isofentanyl pyrophosphate will now condense for the next reaction to form generyl pyrophosphate. The generyl pyrophosphate will now condense with another isofentanyl pyrophosphate. Remember, the general pyrophosphate is come from the 5-carbon molecule. Then the two 5-carbon molecule coming together to form 10-carbon molecule. So general ferrophosphate is actually 10-carbon. So another 5-carbon, which is in form of isophentyl ferrophosphate, will come to condense with general ferrophosphate to form farnesyl ferrophosphate. And farnesyl ferrophosphate will now be a 15-carbon molecule. Are we together? So 15-carbon molecule. Here we have all this stuff. We have the general ferrophosphate, and then we have the general ferrophosphate forming farnesyl ferrophosphate. Now the farnesyl ferrophosphate will now condense with another farnesyl ferrophosphate to form what we call squalene, and squalene now will be 30-carbon molecule, and this process involves this reaction by what we call as squalene synthesis. So this is just an overview of the mobile energy. First, we down to formation of farnesyl ferrophosphate, and from farnesyl we see also other things that can usually be produced using farnesyl ferrophosphate. Now, synthesis of cholesterol from squalene, now it undergoes some oxidation reaction to form squalene 2,3-ethoxide, and this lead to formation of this lanosterol we mentioned. In animals, we have lanosterol formation through some cyclization reaction and multiple step to formation of cholesterol. And we said in fungi we have ergosterol, while in the case of plants we have stigmasterol. Now, effect of cholesterol. Like I said, the cholesterol can be converted to cholesterol ester. This cholesterol ester can be part of the lipoproteins, which is being transported to different organ of the body. And of course, cholesterol also used in the synthesis of fatty membrane. Remember, we also other organs, we said it is used for the synthesis of vitamin D. Now the next one is degradation. Now the degradation also leads to formation of what we call bile acids. These bile acids, we have a primary bile acid and secondary, which we've already discussed this one. So actually the cholic and deoxycholic, and then the secondary we have glycocholic, and deoxylithocholic. We also have this, we talked about the role of these bile acids in detergents, that is in the emulsification of fat. This is simple regulation of bile acid, where cholesterol synthesis. We mentioned the HMG-CoA reductase, the step in the formation of mevalonate, which is being controlled by the action of an enzyme. And the next one is the cholesterol itself, the cholesterol and the cholesterol ester, where we have a feedback inhibition of this HMG-CoA reductase. So when we have cholesterol being produced in high quantity, this cholesterol actually inhibits the HMG-CoA reductase. And actually, accumulation of cholesterol also activates this formation of cholesterol esters. And equally also the cholesterol we have earlier mentioned being packed in form of lipoprotein. They also inhibit the formation of this HMG-CoA reductase. Now the tutorial questions. The tutorial questions, this one's supposed to have been an interactive class. Nevertheless, I have actually been trying to see how I can have time to calm down. Keep on planning, and every time, by the time I'll be there, it's already late or just some few minutes time, so I couldn't make it. But nevertheless, we can go. I will just give you some few hints on how to do. The first question says the first three out of positive fatty acid oxidation follows a reaction sequence closely resembling sequence in the citric acid cycle. Use equation to show the analogous reaction sequence in the two pathways. Look, the first 3 out of 4 steps of fatty acid oxidation. Remember, we talked about 4 steps in fatty acid oxidation. I don't know. or have often this so that to the other PowerPoint talking about fatty acid oxidation. So we can see what we're talking about. Remember, while talking about to be better, we have this. Let me see. Fatty acid oxidation. These reactions. Now the first reaction, which I said, is oxidation reaction, and this reaction catalyzed by acyl-CoA dehydrogenase. I made mention of succinate dehydrogenase, right? Talk about succinate dehydrogenase, and where we said it's a flavoprotein, and we also said this acyl-CoA dehydrogenase is a flavoprotein. It uses FAD, and we said this FAD is like a phosphatic group from these enzymes. So the questions actually want just an equation of this reaction, which is the palmitoyl CoA plus FAD by the action of acyl-CoA dehydrogenase. They give trans delta 2 enoyl CoA, and then the next reaction, which is hydration reaction, and we cite in the case of this, since we have the reaction of fumarase. So in the case of the equation of this one, and you can also give the equation of the reaction catalyzed by fumarase. Now this enoyl CoA hydrate is similar to fumarase, and we have beta hydroxy acyl-CoA dehydrogenase, which is similar to malate dehydrogenase. So this is what this question is talking about. This first step in fatty acid oxidation is closely related. Now we use the equation to show this analogous reaction sequence in the two pathways. So you can put the first reaction, which is, I said, is oxidation reaction. So equally you put the reaction in the fatty acid oxidation in the equation, and now you put the equation of the analogous equation in the TCA cycle, which is catalyzed by succinate dehydrogenase, and then the next one by the fumarase, and the next one by malate dehydrogenase, with the analogous in the case of fatty acid oxidation. Now the next question say, on a per carbon basis, where does the largest amount of biologically available energy in triacylglycerol resides? In the fatty acid functions or the glycerol function? Indicate how knowledge of the chemical structure of triacylglycerols provide your answer. So this one, I think, is much of a direct something, because you are comparing the energy value of glycerol and fatty acids, and that one is quite obvious. You don't need to go that how much energy is produced while metabolizing a glycerol, and how much is produced while metabolizing the fatty acids. And remember, the fatty acid in triacylglycerols, you have even one fatty acid compared to glycerol. You can to compare. The fatty acid is quite higher than this things. So this is just what the question is talking about. So you just mention that there's energy, high amount of this energy. I don't need to tell you the answer, because it's just most of the questions one you see, some of them are just one sentence answer. Some of them are even less than a sentence. That's one press like that. How many tons of fatty acid oxidation cycle are required for complete oxidation of arachidic acid to acetyl-CoA? So this one, I think the most important thing here to know is that how many carbon is the arachidic acid. Just say how many tons. Just say it, okay? So this one doesn't have any distance. The next one say, after a person has ingested large amounts of sucrose, the glucose and fructose that exceed caloric requirement are transformed to fatty acid for triacylglycerol synthesis. This fatty acid synthesis consumes acetyl-CoA, ATP, and NADP. How are these substances produced from glucose? So the question is how acetyl-CoA, ATP, and NADP obtained from glucose. Simple. We'll talk about how this acetyl-CoA is actually coming from TCA cycle. Of course, the glycolysis, the breakdown of glucose to form pyruvate. The pyruvate transported to mitochondria, where it's converted to acetyl-CoA. Then the acetyl-CoA is taken out of mitochondria. So actually, the acetyl-CoA come from this TCA cycle, which come from the glucose metabolism, and the ATP is also come from this same process, TCA cycle, electron transportation, which is all from the glucose metabolism. And NADP, how is this ones? What are the sources of NADP? Remember, we're talking about this same this. So the hexosmonophosphate shunt, which is also found from the glucose metabolism, is used to produce NADPH. So this is just the important things just to mention. Now the next question says, in the condensation reaction catalyzed by beta-keto acid ACP synthesis, and this is fatty acid synthesis, a 4-carbon unit is synthesized by the combination of 2-carbon unit and a 3-carbon unit with the release of CO2. What is the thermodynamic advantage of this process over one that simply combine two carbon units? This question talking about the thermodynamics, the energy. Remember, we are talking about bond breakage and bond formation. We mentioned that some certain reactions, depending on the energy requirements, if you want to form a bond, you must have an energy. And where does this energy comes from? The energy comes from the breaking of bond. So the energy released by breaking a bond can be used in the formation of bond, depending if that energy is enough. If the energy is not enough, then you have to use ATP, or you have to use a source of energy. You have to use a source of energy to do that. Breaking down of another maybe bond that has a high energy bond, then it can be used for this. In this one, where we have this action of beta-keto acid ACP synthesis, we have this malonyl-CoA, and remember the removal of the CO2, which was added by acetyl-CoA carboxylase, is removal of that release a lot of energy, and that energy is used for the condensation of this acetyl group attached to the tail of the ACP to the malonyl-CoA attached to the ACP. So the energy is actually coming from the removal of this CO2. The energy involved, this is what is used. But normally, when you, for example, you are using 2-acetyl-CoA, because this question says over one that simply combine two carbon units. Let's say two carbon units like acetyl-CoA and acetyl-CoA. Of course, the condensation of acetyl-CoA, like we've seen in the case of ketogenesis, and also in the first step in the production of cholesterol, we've seen that formation of acetyl-CoA. For the 2 carbon to come together to give acetyl-CoA, we have the removal of coenzyme A, and that energy of bond breakage that lead to release of coenzyme A is what actually drive the condensation reactions in the formation of this carbon-carbon bond. But assuming is acetate coming together, that's possible. Then external energy actually has to be used. So that's just the thermodynamic. The thermodynamics lies behind the removal of carboxylic group, which is added by acetyl-CoA carboxylase. Now the next question says the biosynthesis of palmitalate. It is a common unsaturated fatty acid with a cis double bond in carbon 9 position. It uses palmitate as a precursor. can this be carried out under strictly anaerobic condition? Explain. Now, this explanation is just simple and direct, because the enzyme basis on synthesis, we made mention of this enzyme desaturase, and this desaturase, I said, the fatty acid desaturase, I said, is mixed function oxidase. Be it being a mixed function oxidase, I said there are quite a number of reducing molecules involved in the reaction, and we said the final electron acceptor here is oxygen for this enzyme. So assuming oxygen is not there will be no final electron action. The enzyme cannot actually do this desaturation reaction, removal of these electrons. So only it's here is you have the saturated fatty acid in the presence of this oxygen, by the fatty acid desaturase actually lead to formation of this monounsaturated fatty acids. This oxygen is being converted to water, even though we have this electron carriers as part of the prostatic group, the center of the enzymes. The final electron acceptor here is oxygen. So the questions is just trying to ask the role of oxygen under anaerobic condition. Of course, oxygen is not there, so this enzyme cannot actually do this saturation reaction. Now the next question says, use a net equation for the biosynthesis of tripalmityl glycerol, that's tripalmitine, from glycerol and palmitate to show how many ATP are required for molecule of tri. So this answer, I don't think I need to go that, because you talk about the synthesis from glycerol, formation of glycerol 3-phosphate, how ATP is actually being utilized, and then you from this side, before you come to the transfer of acetyl-CoA group, how does the fatty acyl-CoA, how does the fatty acyl-CoA even produce? Because in the formation of the fatty acyl-CoA, remember where this ATP is used and converted to AMP, and we said this process actually used to ATP directly. So formation of fatty acyl-CoA to ATP. So how many fatty acyl-CoA in triacylglycerol? Like 3. So from there you even have a 6 formation of fatty acyl-CoA. Now the transfer, and the transfer doesn't need energy, and formation of a glycerol 3-phosphate. Okay, so the energy is there, and here we have a 6 energy, so 6 ATP. So I think you can just do the calculation and tell how many ATP is actually required for this process. The next question says, differentiate between TAG synthesis in the small intestine and in the adipose tissue. Yes, this is simple and direct, I think. Remember, we're talking about the lipid absorption. We said something about monoacyl glycerols. Monoacyl glycerols is what is used for the synthesis of TAG in the small intestine, while in the case of adipose tissue in the liver also, fatty acid or triacylglycerol synthesis actually started with either the glycerol, where we have the glycerol kinase forming glycerol 3-phosphate, or it started from dihydroxyacetone phosphate. Hydroxyacetone phosphate dehydrogenazed to form glycerol 3-phosphate. Okay, so this one started from sucrose, while this one started from monoacyl glycerols. Mostly two monoacyl glycerols. I think one of the two difference, or one of the major difference, I think. Now when you read, you can get more also. Now the next one say lipid digestion and absorption heavily depend on fatty acid metabolism. Just by this one, I think I've answered it while discussing about this cholesterol metabolism, information of bile acids, how cholesterol is being converted to bile acid, and how bile acid actually play a very important role in digestion and absorption. Imagine bile acid is not there, so digestion and absorption, as explained based on the Backstrom hypothesis, this will be very difficult, of course. Now the next question says, based on your knowledge of biochemistry 262, suggest a predominant pathway through which a ruminant animals meets their energy demands. So this one has also mentioned it while discussing this, this microbial action in the small intestine, ruminant in the ruminant animals, in the rumen. So in the rumen we talk about these microbes and how these microbes actually, even when you take a food or animals take a food, this carbohydrate base, how the glucose is being produced in the small intestine, that they actually being absorbed as glucose. Mostly this glucose are actually broken down by these bacteria, and what the bacteria produce, the volatile fatty acids, and also even the part we talk about in the ruminant animals, even the long chain fatty acids are broken down into these volatile fatty acids. So all of them taken in this small volatile fatty acids. These volatile fatty acids, some of them will be using directly formation of acetyl-CoA. Some will go through prophenyl-CoA and prophenyl-CoA. Remember how can prophenyl-CoA can actually be utilized in the body. So you can make sure you remember that one. How prophenyl-CoA can be utilized in formation of TCA cycle intermediate, and how that can, if possible, can be used for synthesis of glucose. And the acetyl-CoA, like I mentioned, and also this beta hydroxybutyrate, the ketone metabolism. So you can suggest from there, which is mostly actually fatty acid metabolism, because fatty acid metabolism is altogether ketone body synthesis, and ketone body utilization is to do with fatty acid metabolism as the main source of energy, predominant pathway for energy production in ruminant animal. Now the next question say double membrane structure of mitochondria is very important in energy production during electron transport chain and oxidative phosphorylation. Explain. So this one also doesn't need any distance. Double layer membrane double layer membrane, where we said in the first membrane, the outer membrane is the porous. It's due to the presence of these porins which allow the movement of substances in and out to the intermembrane space, and we've seen the concentration of proton being high in the intermembrane space compared to the mitochondrial matrix, and how these molecules strictly moves into the matrix under strict condition by the action of those transporters, and how this leads to formation or building of this proton potential, protomotive force in between the two membranes or between the two compartments, lead to energy production. So what important to this? To what important is this for energy production? The next question say malonyl-CoA is or not a basic unit in fatty acid synthesis. Discuss. So this one you can also answer it. Remember we said the fatty acid synthesis, we have acetyl-CoA coming together to give fatty acid. But what we have seen is the malonyl-CoA actually is coming. But the question is, the malonyl-CoA is formed from acetyl-CoA, and this malonyl-CoA involves addition of carboxyl group, and this carboxyl group finally ended not as part of fatty acids. So I think I should leave you there. You know where the answer comes from. So you discuss whether it is or it is not, and you give your reading. Write the net equation for the biosynthesis of palmitate in the rat liver, starting from mitochondrial acetyl-CoA, cytosolic ADP, ATP, and those. These ones I don't know. It's a direct question, so has no explanation. Need no explanation. Now the next question say the first two reactions involved in the formation of HMG-CoA and ketone body formation are similar to the first two reactions in cholesterol synthesis. Identify the two enzymes symbol. Simple. Which is direct. I think I also talk about it while discussing these two. So I discussed these two before. Discuss this. Now the next question say the concentration of ketone bodies, especially beta hydroxybutyrate, tend to be higher in ruminant animal than in single stomach animal. Just to buy all this also I'll give quite a number of explanation on this. Remember how we say this beta hydroxybutyrate, this is butyric acid as volatile fatty acids directly after absorption can be converted to beta hydroxybutyrate. Simple. Ruminant animal, what we have as a source of energy mostly is volatile fatty acids. Volatile fatty acid actually more than 80 to 90% of the energy meet of the energy requirement for ruminant animals, and butyric acid is one of them. So the concentration of butyric acid is high in ruminant animals, and once the butyric acid get absorbed into the system, it's converted to beta hydroxybutyrate. And you can justify this smelling of ketone body from its ruminant animals. When they breathe, you feel the smell or something like that. Because we said the ketone bodies give one kind of distinct smell odor. That's why even the diabetic individuals, at times when they are diabetic situations, you feel this odor coming out from their body as a result of the accumulation of ketone body. Now the next question say identify 3 differences in lipid digestion between ruminant animal and single stomach animal. 3 differences in lipid digestion between ruminant and so this action of what do you call it? This action of intestinal microbes. So we have the action of microbial lyphases, which you don't have in single stomach animal. We have the action of this formation of volatile fatty acids, and we have this what do you call it? We have this dehydrogenation reaction. Hydrogenation reactions. We have hydrogenation of the fatty acids, where we said even there are these fatty acids that are unsaturated. The unsaturated fatty acids usually get hydrogenated, so they are converted to unsaturated fatty acid before they get absorbed. Now the next reaction say explain how beta oxidation of fatty acids differ from alpha and omega. Remember I gave you example with assignment on that, and this is only the assignment I didn't mention, so you must read this. This is compulsory for you to read. The next says fatty acid desaturates is most likely to be less active in ruminant animal compared to single stomach animal. Fatty acid desaturates. This is just an assumption that assuming that the energy, the fatty acid is actually coming from diet. So this fatty acid coming from the diet, most of them are saturated fatty acid. No, I think there's something wrong with this question. So I think this question is there's something wrong with this question. Yeah. So I think I will modify the question. So, this is what I meant. We're talking about the hydrogenations that occur in the intestine before absorption. So most of the fatty acids coming from these animals actually is from the ruminant animals is saturated fatty acids. So I think something is missing in the questions. So I'll correct the questions and send it back, this particular one. But this is what the questions is trying to talk about. So it's only the questions does not comes out clearly. So if you have anything missing or not understood, I think you can have a direct contact with me from now to Saturday. On Saturday we're going to have our test, and this will give you a hint of what kind of questions are you expecting. And believe me, by the time you try it and make sure you understand how these questions were answered, how you can address these questions, you will not have a problem with the test questions I would have. Because you will see something like this, and if there is something out of this, will be much simpler and direct than these things. So no matter how the questions actually been manipulated, they will not be manipulated more than this level. So if you have the highest complexity at this level, then the ones you have more direct that are not as complex as these ones. This is what you are expecting. So if you have any question, you can meet me directly, or you can follow through the class rep and have access to me. We are always available, because actually you can meet the class rep and find a way of chatting me up if you could not meet me, because sometime the time you'll be available and the time will be also available in office might differ. So just make use of any available opportunity so that you can have your problem before the distance. You can have your problems taken care of. Please, I hope this satisfies almost all what is expected for me. For the revision of other things, I felt there is no much things needed for the revision actually. These ones should be enough. The other ones that you're expecting us to go through is just saying the same thing. There is no much where these logics and the connections comes in. The reasonable things you can connect A and B, or this pathway and this pathway. What I picked here, quite a number of them is what I picked here. I said it here. So we don't need much to talk about in the other aspects. So this serves as the revision class, and it's also serve as the tutorial class, the tutorial questions. So see you guys. I hope this one will solve most of the issues you might have seen in this course. And by now this course will be the simplest course this semester, if not the only and the simplest one. Bye. See you.